Everyone, as you all file in here, I think we're going to get started so we can try to stick to our times as closely as possible. Welcome to our final day of Think Tank. We are going to get started with another Beacon Research Award session where we will hear from our remaining 2022 Young Investigator Awardees, including our patient-centered clinical research Young Investigator Awardee. So if I could have both of our speakers please join us on the stage. And I'm also introduce again Dr. Peter Clark, who's been the chair for the past 2 years of our Young Investigator Award Scientific Review Group, and Dr. Sarah Suka, who's also been the chair for the last 2 years of our patient-centered Clinical Research Young Investigator Award Scientific Review Group. They are going to introduce our awardees before we get started with their presentations. Again, each awardee will have 15 minutes for their presentation, followed by 5 minutes to take questions from the audience, so please make use of the microphones on either side of the room. A huge thank you to our chairs, Peter and Sarah, and congratulations to our awardees. And with that, I will hand it over to Peter.
All right, good morning, everyone. Welcome to our second round of awardee presentations. I'm looking forward to hearing some of the exceptional work that we're going to hear this morning. Again, my thanks to the Beacon staff and the entire study section for helping put together the best work and to bring it for you today. So this morning we're going to start with Dr. Weisi Liu, who is a postdoctoral fellow in hematology and oncology at Weill Medical College of Cornell University. Dr. Liu is a senior postdoctoral fellow in the Faltis Lab at Weill Cornell Medicine. He is focusing his efforts on studying the mechanisms and downstream effects of ABEBEC-induced mutagenesis in urothelial carcinoma and developing therapeutic strategies to target the mutagenic fuel of bladder cancer cells. Dr. Liu has published in peer-reviewed journals and recently received a training grant from Anvil Cloud Credits Continue Program, or the AC3, from the National Human Genome Research Institute. And his title for today's talk is Synthetic Lethal Strategies to Limit Tumor Evolution Driven by ABEBEC Activity in Urothelial Cancer. Dr. Liu.
Thank you, Peter's introduction. Good morning, everyone. So my name is Weisi Liu. Upgrade a little bit. I'm right now the instructor in the Dr. Faltis Lab at Cornell Medicine. So it's a great honor to stand here to present our current work named Targeting MMEJ by Inhibiting Polycyta in the Urothelial Cancer with ABEBEC3 Activity. So brief introduction ABEBEC3 ABEBEC enzymes. So these enzymes have 11 members in humans, and these enzymes deaminate the cytidine in the preferred TC motif in the single-stranded DNA and RNA. So they will result in the C2T or C2G mutation in the DNA due to the DNA repair mechanism or CTU editing in the RNA. So this enzymes, the main functional is immunorelated. So they were involved in the virus restriction and the diversity of our antibodies. And also they are related to the metabolism using their RNA editing function. By detecting the mutation footprints in our human cancer genomes, ABEBEC3 induced mutations are highly enriched in the multiple cancer types, including the bladder cancer. That's indicated that the cancer cells hijacked these enzymes as a mutagenic fuel to increase the tumor evolutions. Our peers' paper have been found that the ABEBEC3 induced mutations are highly enriched in the chemotherapy resistant cancer patient samples. And recently, the paper found that the therapy itself can trigger the ABEBEC3A expression and drives the evolution on these survival cells. That would be sad because that means every time you give the patients with the treatment, and meanwhile you will increase the opportunity to get a resistance to this treatment because ABEBEC induced the tumor evolutions. So what can we do? Is there a small molecular inhibitor can target the ABEBEC3 enzymes? The answer is no. Unfortunately, there is no currently inhibitions in the market can specifically target ApoBac3 enzyme. So is there an alternative way to target the downstream, the DNA repair mechanism to restrict the ApoBac3 activity and also illuminate the cancer cells? So to address these clinical questions, we need to answer first, does ApoBac3 induce the lethal DNA damage in our cancer cells? Second, what's the preferential downstream, the DNA repair mechanism for the ApoBac induced the lethal damage? Number three, is inhibition this repair mechanism is synthetically lethal in the cancer cells with ApoBac3 activity? So to answer this first question, we generation a cell line model using in the 5637 cells using the doxycycline-induced ApoBac3A. So we choose ApoBac3A because the previous paper found that ApoBac3A is the major contributor to the ApoBac mutation in the bladder cancer. So we turn on the ApoBac3A for the 24 and the 48 hours, and then we check the replication stress marker and the double-stranded DNA break markers. As we're showing here, you can see the tumor cells when the exposure to the ApoBac3A, the CHK1 and RPA is highly activation in the cells. That's indicated that the cells have the replication stress. And also the marker of the ATM, the CHK2 and the gamma H2X also highly activated in the cells. That's indicated the double-stranded DNA break. So that's data indicated that the ApoBac3A can cause the replication stress and the lethal DNA damage, double-stranded DNA break. So how the ApoBac3A induce the replication stress since they just deaminated the Saidian as a single-based mutation? So to answer this question, we're using the DNA fiber assay to identify the impact of the ApoBac3A activity on the replication fork. So this DNA fiber assay using two different thymidine analogs. So these analogs can integration into the DNA when the replication process. And then we can using the antibody to specific labeling this DNA fragment. When we compare the ratio between these two fragments, as we're showing here in the right, so you can judgment the replication status is ongoing or is terminated. So we did this experimental in our cell line model, as we're showing here with the represented image and the quantification data. It's clearly that the ApoBac3A decreased the ratio of these two fragments, indicated that the replication fork is terminated, and then might be the reason triggering the replication stress and accumulation of the double-stranded DNA breaks. So right now we know ApoBac can cause the lethal damage. So what's the preferential downstream, the DNA repair mechanism for these damages? Before I answer these questions, we need to know that there are the three major double-stranded DNA break repair mechanisms in our cells. One is called the classical NHEJ, another two is HR repair and MMEJ repair. So since the ApoBac-induced double-stranded DNA breaks happens in the replication phase, so we focus on the HR and the MMEJ repair because these two mechanisms is predominant in this phase response to the double-stranded DNA breaks. As I highlighted here, so the single-stranded DNA overhangs at the break's end, binding by the RPA is the essential steps to make the decision choice for the repair. So when the single-strand DNA overhangs, binding by the RPA, and then the RPA replaced by the RD51, so the repair will go to the HR repair using the cysteine chromosome as a template. But if they have the limited the RPA displacement, so they will exposure the DNA to overhands and then promote annealing using the microhomologous sequencing, and then the repair will go to the MMEJ mediated by the polytheta. Of course, the MMEJ repair is the error-prone repair process, so that's why we'll create the unique mutation footprint called the microhomologous mediated deletions in our genomes. So is that ApoBac3A can significantly increase the microhomologous mediated deletions. So we're sequencing our cells, the cells model. So these cells already exposure to the ApoBac3A over 2 months. And then as you can see here in the left, the ApoBac3A expression cells have significantly higher number in the indels, indicated that the error-prone DNA repair mechanism is recruited. And also after the classification definition by the Cosmic, we find the microhomologous mediated deletions also significant in our cells. So to validation our experimental finding in the human samples, we did a whole genome sequencing in our patient cohort. So as is showing here in the left, is there a significant correlation between the AppleBax-induced mutation burden and the microhomolog media deletions. And we also found that this significant correlation is not only happen in the bladder cancer, it also happens in another cancer types when we're using the TCGA pan-cancer whole exome data as we're showing in the left. So this result indicated that these two mutation footprints consistently occurs in the human cancers. So we know, right now we know that MMEJ is preferred to respond to the double-stranded DNA break caused by the AppleBax3A. But why is MMEJ? Why not is the HR repair? Does the AppleBax3A will direct involve into this decision maker? So as I mentioned before, the RPA binding to the single-stranded DNA overhand is essential steps for the choice, repair choice. And also the AppleBax3A can binding the single-stranded DNA overhands. So we have some hypothesis that AppleBax3A can displace the RPA, resulting in the expression of microhomolog sequencing and promote MMEJ repair mediated by the polyceta. So to answer these questions, we designed a biochemical assay as we're showing here. We're using two oligos to mimic the break ends. So the longer one on the 5 ends will label with the fluorescence marker for our detector. And also the 3 ends will have the microhomolog sequencing, and in the middle is the motif for the AppleBax binding. And then we mix these oligos with the A3A, RPA, and the polyceta proteins. So in the reaction, if the AppleBax3A overcompete with RPA, so they will exposure more microhomolog sequencing, and then they will annealing, and then the MMEJ polyceta will involved in to generation the longer annealing products. But if the RPA is overcompete with AppleBax3A, so the microhomolog sequencing will be protect. Sorry about that. Where we go? Okay. So when the RPA overcompetes with the AppleBax3A, so RPA will binding the single-strand DNA overhands and protect the microhomolog sequencing. So they will inhibit the MMEJ repair mediated by the polyceta. So when we do the reaction and run the gel, so here is the gel image. So the upper band is the annealing products, and the bottom band is the unannealing products. As we highlight here on the left, you can see when we added the polyceta into the reaction, so they were generating more the annealing products as we expect. But when we added the RPA into the reaction, it's highlighted in the middle, so the products were decreased because the RPA protect the microhomolog sequencing at the 2 ends. And when we added the AppleBax3A into the reaction, the annealing products reversed, significantly increased. So here is the quantification outcome. That data indicated that the competition between the AppleBax3A and the RPA for the single-stranded DNA overhands on the 2 ends, resulting in the annealing of the microhomolog sequencing and promote to the preferential to the MMEJ repair. So right now we know the MMEJ repair is the downstream double-stranded DNA break repair mechanism to the lethal damage caused by the AppleBax3A. So is there the inhibition, the polyceta, the key protein in the MMEJ is synthetic lethal in the tumor cells with AppleBax3 activity? To answer this question, we go back to our cell line model. So we knock down the polyceta using the shRNA. As we're showing here in the left, it's the knockdown efficiency using the qPCR by the 2 different primer sets. And then we turn on the A3A. As we're showing here in the right, with the AppleBax3A increase their expression level, the polyceta knockdown cells are much more sensitivity to the AppleBax3A induced toxicity compared to the SH control cells. So we want to test, is that this effect also can be used in the clinical? So we recruit 2 different polytheta inhibitors to identify the synergistic effect between the ApoBac3A expression level and the polytheta inhibitors. So we recruit one drug called novobiocin. So it's targeting the ATP binding domain. Another drug is called ART558. It's targeting the polymyxin domain. So these 2 drugs right now is in the clinical trial. So we performed the cross-experimental with the dose curves of these 2, with the doxycycline, which indicated the ApoBac3A expression level. It's on the x-axis, and also the concentration dose curves with the drug, the polytheta inhibitors. Here is the synergy map. So the red area indicated the concentration of these 2 effects are synergistic, and the green one indicated the opposite direction. So we're using the ZIP model to calculation the synergy score, and then these 2 scores indicated that there is a synergistic effect between the ApoBac3A expression level and the polytheta inhibitions. So we're still working on the experimental validation our synthetic lethality in our organoid model and the patient-derived xenografts model. I think the missing one slide, but it's fine. So in conclusion, so basically we find that the ApoBac3A can induce the lethal double-stranded DNA breaks in the cancer cells. And the second, we find that the polytheta-mediated MMEJ is the predominant downstream DNA repair mechanism response to this lethal damage. And also we found that targeting the polytheta is synthetic lethal in the cancer cells with ApoBac3 activity. So to end, thank you for my mentor, Dr. Feltus, their support, and also all the members in the Feltus lab, and also thank all the collaborations for their efforts. And of course, thank you to the support from the Beacon. So I'm happy to take any questions. Thank you.
So I had a quick question regarding, and I don't know the DNA repair pathways perhaps as well as I should, but we know that in bladder cancer, and we've heard about a variety of different germline mutations, BRCA1, BRCA2, ATM, et cetera, et cetera, et cetera. Do you have any sense yet of how those types of mutations could interact with this? Because, in a lot of these cells, there's going to be more than one aberration. So how do those 2 interact, and how do you think that's going to affect your results or impact how you use this ultimately clinically?
That's very great questions. So currently, these 2 inhibitors for the MMEJ repair is tested in the breast cancer with the BRCA mutation. And also, we know they have patients in the bladder cancer also have the BRCA mutation. So I'm thinking if the patients have the BRCA mutation and ApoBac's high burden, high activity, which will be more sensitivity to these drugs. So we're still using this filter to try to figure out in our organoids. Mm-hmm. So we classify them with the organoids like ApoBac mutation, if they have the mutation in the BRCA or not, or another DNA repair mechanism to test their response to the drugs. So we're still working on this.
Okay, because I would anticipate that the mutational profile within the different organoids that you're testing might very well influence their response to these.
Yeah, exactly.
Yes. And actually that offers a potential opportunity to actually have predictive biomarkers developed as part of this process, which is actually really exciting. And it could be a first, really, in many respects.
Yes, exactly.
That's awesome.
Thank you.
Go ahead, Bill.
Oh, sorry. You're looking over there.
Just making sure there wasn't somebody else, that's all.
So I think Charles Swanton's group has published that, at least I believe primarily in cell lines, there's more episodic expression of ApoBac, and so I'm curious what your thoughts are about in vivo human tumors. Is the episodic expression reflected in that? And if it's truly episodic, then does that prohibit your synthetic lethal approach if there's not actual ApoBac3 activity ongoing in a tumor?
I just rephrase your question. So you're thinking because our model is using the overexpression system. So you're thinking in the patients, the expression level will not be that higher enough to create more damage. Is that correct?
So there was a paper that suggested that the expression of APOBEC, albeit in cell lines, is episodic, meaning it's not always constantly mutagenizing, but it's this sort of intermittent activity that's driving the mutagenic process, which is perhaps reasonable given the the replication stress you're showing and the inability of cells to actually tolerate that long term. So what I'm curious about is your thoughts, if this is true. So I think we actually, if anybody wants to take it on, I think we should actually look at human tumors, bladder tumors over time and see if there is truly APOBEC activity that's differentially regulated over time. But I don't think we actually have, maybe your replication stress assay is nice, but I was thinking the classic deaminase assay isn't really that sensitive, at least in our hands. And so
Okay.
I'm curious whether you think this is a problem for your Pol theta inhibition
Yeah,
strategy.
that's a great question. So, first we test in our organoids with the APOBEC mutation burden to predict is there a reaction, the response to this polytheta inhibitions. And then meanwhile, as I show in the paper, the therapy can also induce the APOBEC A3 expression level. So this drug is the EGFR inhibitors. And in their paper, they showed clearly that when you give the cells with these inhibitors, they will cause the double-stranded DNA break using the H2AX markers. And then when we knock down the APOBEC3A, the double-stranded DNA markers decreased. So that means, if we also will, that's good chance if we do not see any good response when we just test in the organoids, we might be a combination with some treatment. So that one maybe after this treatment, they trigger in this expression level, and then we can follow up with the polytheta inhibitor. So we can use in the combination with these two drugs in a clinical trial to see the patient's reaction, and also you can enlarge the response with the drugs, also limited the evolution to the residents.
Yeah, and the other one I thought is, it's clearly like a very nicely upregulated by interferons. So, maybe the steroidin adds, whatever you call it, the add interferon, that kind of thing may also be a nice adjuvant. So
Yeah.
very nice work, though, nonetheless. Thank you.
I echo Billy, great nice work. Congratulations to you and the team and Bisoi. I was actually looking at, recently there was a paper in lung cancer looking at APOBEC signature, the mutagenesis signature, correlating with prognosis and potential response to immunotherapy. Any comments about, based on the mechanism we nicely showed, prognostic role and potential predictive value, just following up Peter's question, in response to platinum-based chemo or immunotherapy, or other therapies in bladder cancer? Again, great work.
I think we're still working on this. So it's good to using this marker to detect the reaction with the chemotherapy as a biomarker. But considering the APOBEC will cause tons of the evolutions, so it might be harder to using all the burden to reaction the chemo, or specific target, say this will cause the resistance or not, because APOBEC create a lot of mutation. Some may be just a passage mutation, some maybe it's a driver. So we still need to working on to identification which one is the driver to the chemo resistance, and then that will might be improve our predictor marker to predict the reaction to the therapy. It may be harder just using the APOBEC or the mutation burden. That's my thinking.
Thank you.
Thank you.
Until we have an effective inhibitor, are there certain agents that are less likely to induce more APOBEC activity? For instance, taxanes, which are microtubular agents, versus alkylating agents, which cause double-strand breaks. Is there a hierarchy of known chemotherapeutics or other drugs that are more likely to induce this accelerated activity?
That's a great question. I think it could be, because the chemotherapy also will create a lot of the DNA damage, but also the issue is when you create a lot of the damage by the chemo, they also will create opportunity for the APOBECs, for the single-stranded DNA break, right? So
Right.
that may be also the chance we can combination with the chemo with this polytheta inhibitors.
I was wondering, since your group is published on the induction of APOBAC activity by chemotherapeutics, whether or not there's a hierarchy of drugs more likely to induce it than others. A listing of known chemotherapeutics and their ability to upregulate APOBAC. Oh, very good. Thank you.
Yeah. Thank you.
Great talk. I'll ask for Bishoy. I'll ask if it's true, what does it imply question. So if there's typically thousands or maybe hundreds of thousands of APOBAC lesions in a typical bladder cancer. So does that mean each site of APOBAC alterations was a double-stranded break, like prior to its activity, prior to the APOBAC I mean, that means that there's like many, like, I guess, did we realize that there's that many double-stranded breaks prior to a APOBAC mediated mutagenesis, or are there potentially other ways that APOBAC may have activity in the absence of a double-stranded break?
I think I'm not fully clear with the question. So you want to ask, is that another mechanism?
I guess the real question is, does APOBAC3 activity, at sites of APOBAC3 activity, are they always preceded by a double-stranded break, or can APOBAC work in the absence of a double-stranded break?
That's a good question. So as B.D. King said, the APOBACs need high expression level. That will cause double-stranded DNA break. So they need to accumulation certain of the mutation burdens that will stop the replication fork. So we have another mechanism can bypass this AP sites caused by the APOBAC. So they will cause the mutation, single-base mutation, but still can go through the replication force, which may not cause double-stranded DNA break. So that's why this therapy or the treatment need more APOBAC activity to create a double-stranded DNA break. But in another side, so if you give the chemo or another DNA break reagents, they will create more double-stranded DNA break. In our experimental, we're thinking if that's the case, so you get more break ends, the APOBACs also will be involved in this decision choice. So that maybe also will be the sensitivity to the polytheta inhibitors.
So you're saying agents that cause double-stranded breaks may induce APOBAC3 activity then?
I think so.
So radiation and other, DNA damaging agents would you might hypothesize would have a similar APOBAC3 inducing activity then?
Yes, so that's with paper our mentor published in the Nature Genetics said the chemotherapy resistance, the patients have highly enriched APOBAC mutation burdens.
Okay, thank you.
Thank you.
Good morning, everyone. My name is Sarah Sutka from the University of Washington and the Fred Hutchinson Cancer Center, and I have the distinct honor of chairing the SRG for the Bladder Cancer Advocacy Network Patient-Centered Clinical Research Young Investigator Award. This award is unique in that it supports early career investigators and researchers who are investigating transformational, patient-oriented research. We seek proposals that span the critical topics such as access to care, quality of life, patient-reported outcomes, access, equity, and health disparities, as well as comparative effectiveness research and survivorship. It is my true honor, and I'm delighted to present the 2022 awardee, Dr. Ava Saidian, who's going to present to us today on her work looking at disparities in patient-reported outcomes among Spanish-speaking Latinx patients with bladder cancer. Dr. Saidian recently completed her fellowship at the University of California, San Diego. She earned a BS previously at the University of Texas and her medical degree from the University of Kansas School of Medicine, before she was inducted into the Alpha Omega Alpha Honor Society. Prior to that, she completed her residency at the University of Alabama at Birmingham, and we're truly excited to hear about your work today. Thank you.
Thank you for the introduction. So, like Dr. Suka mentioned, I'm going to talk to you today about our study titled Disparities in Patient-Reported Outcomes Among Spanish-Speaking Patients with Bladder Cancer. So I started this project as a fellow at UCSD, and I hope to continue it as a faculty member at the University of Tennessee. So just some background information. The incidence of bladder cancer in the Latinx population is lower than the general population. However, this group tends to have worse survival outcomes. The details surrounding their survivorship is unknown. There's not a lot of studies or data related to survivorship in these bladder cancer patients. There have been other widespread studies that have found that even minor language barriers can affect the quality or type of care a patient receives. An example of this in bladder cancer was a study done in Texas by Dr. Wu's team, where they looked at just different outcomes related to bladder cancer care. They found that Latinx patients, when corrected for other variables, were less likely to receive a continent diversion. So neobladders or a pouch were more likely to get an incontinent diversion. There's obviously a lot of factors that lead to that, but one of the major things that they postulated was that a lot of this was due to language barriers or things that relate to language barriers, such as inadequate patient counseling by the physician, or just not understanding patient-centered preferences, as they had a provider who had different, who might not understand their unique cultural beliefs. So we know that there are disparities that exist in cancer care, not just bladder cancer, but other cancers between different ethnic groups. However, disparities in health-related quality of life outcomes in the Latinx population compared to the non-Latinx population has not been well described. So that's what we sought to do. Given we're in San Diego, we have a very large Spanish-speaking population. So we designed our study around three aims. We wanted to establish if there was a true disparity in quality of life outcomes using patient surveys. We wanted to interview patients to better understand their perioperative experience, and then we wanted to translate a decision aid into Spanish so that we could hopefully start to bridge the gap in care in this population. So we used a number of different surveys to try to establish any disparities in this group. Before I get into all the surveys, I do have to mention a huge problem that we faced was a barrier to patient recruitment. I was kind of naive, and I thought, oh my gosh, we have so many Spanish-speaking patients. This is going to be great. We're going to solve all the world's problems. However, you can't do that if patients aren't participating in research. There were a lot of different barriers to recruitment. Some things that stuck out were not having phone access or email access, which is how we did the surveys, or their phone access was inconsistent. They were elderly patients who their main contact was their child, who worked all day, so we had difficulty just communicating with the patients, or they were just not interested because they felt like it was a large effort and they didn't want to burden their family by participating in the study, which is all very understandable. Okay, so anyways, back to the surveys. So the first survey that we use is the Bladder Cancer Index. It's 36 items, and it covers 3 different domains: urinary, bowel, and sexual health, divided into function and bother. I'm sure all the clinicians might be familiar with this. A higher score relates to a better health state. So again, we had a super low response rate in our Spanish-speaking population, so all of our data was not statistically significant. However, there were some trends. Primary English-speaking patients tend to have higher scores, so a better health state in terms of urinary function, being less bothered by any urinary symptoms. Same with bowel function, and they were less bothered, and though they reported worse sexual function, they were actually less bothered by it. Again, none of it's statistically significant. Hopefully we can make some more concrete statements once we recruit more patients. So our next survey dealt with body image. Some background on that. We know anecdotally as a physician, we can only imagine the changes to the body that happens as you undergo bladder cancer treatment. There was a prospective study done recently looking at health-related quality of life outcomes between patients who received neobladder versus ileoconduit. They found that there weren't actually that many differences in the long term, except for in a couple in two domains. One being in body image. They found that patients who received an ileoconduit had worse or a greater decrease in their self-reported body image compared to those with a neobladder. So they gave them the survey baseline right before surgery and then at different time points afterwards. And the patients who received an ileoconduit compared to a neobladder kind of had lower scores in the end. The way that they did this was using a survey that had just 3 questions relating to body image, which you can see here. We wanted to use a tool that was a little more robust, and so we used the body image scale. It's a 10-point survey relating to body image. It was initially developed for breast cancer patients and has only been validated in breast cancer and colorectal cancer patients. So we thought this was also a great opportunity to not only see if there was a disparity between these different ethnic populations, but to also validate this survey so that other clinicians could use it in their studies. For example, like single versus multi-port. Do patients actually care about cosmesis? Is there a difference? Things like that. So a higher score correlates to a self-reported worse body image. Again, we had super low responses from the Spanish-speaking group, and none of our outcomes were statistically significant. But we did begin preliminary validation. We established reliability, face and content validity. That's all just boring stats talk, but basically those are weak forms of validation. What we really want is clinical validation, so other people can use this, which we started, we've begin starting that validation process. And as we get more participants, we should be able to establish validity of this survey and being able to tease out how patients feel about their body image based on different subcohorts. And then our last one was satisfaction with decision. Again, we gathered a lot of preliminary data on how patients felt about their decision to have an ileoconduit versus a neobladder, and then break down on different subgroups. There was some small differences. English-speaking patients were happier with their decision. Neobladder was, just barely slightly higher. Again, none of this was statistically significant. So the second part of our study was patient interviews. This was a really unique experience. A lot of what we learned is probably stuff that patients, patient advocates, and patient family members know. As a clinician, I personally don't get a lot of time to talk to patients about their quality of life. Our 15-minute visit is usually spent talking about the state of their cancer, our next treatment options, going over any issues they might be having, but not really quality of life. So getting to spend an hour with patients just talking about their quality of life was, really interesting, and I hope some of the things I learned can help other people with their patients. Again, we had no Spanish-speaking patients that were interested. We're still trying to recruit to see if there are any, different themes that emerge from our Spanish-speaking population. But our interview was divided into 3 different phases, so preoperative, perioperative, and then long-term phases. In the preoperative phase, we asked, how did patients feel going into surgery? Did they feel like it was the right decision? Did they have confidence in their team? Everyone, most people felt that they felt confident it was the right choice to undergo surgery, and they said that this confidence came from not only family support, but mostly from their urologists and their medical oncologists. So just having confidence in their team gave them confidence into going into surgery. Otherwise, the patients who didn't feel confident in their surgery decisions felt like they had, they were happy they went through the surgery and that they're doing okay with their cancer, but they just seem to dwell a lot on, should I have done this? Was this the right thing? Did I have enough information going into it? Then we asked patients, what resources did you get preoperatively that helped you decide between an ileoconduit or a neobladder? So there were a couple patients that were told that they should get an ileoconduit just based on their comorbidities and their cancer. Otherwise, most patients said that they just received a handout describing the differences, or they saw an ostomy practitioner who, helped them better understand an ostomy. Otherwise, it kind of ranged from patients not really knowing what they were going into, just blindly trusting their physician, versus others doing research on their own. And I think what was really interesting was that videos were really helpful, which makes sense when you think about it, but in a clinic visit, getting those resources to a patient is not always at the top of your head. So I really like what one patient said, which is, I watched a YouTube video of someone changing a bag. They made it look pretty easy. I saw it before surgery, and it helped me feel very confident seeing someone do it. It was more helpful than the cartoons or the people with fake plastic stomas on. I didn't know people did that. That's interesting. So immediately post-operative, we asked patients, was there anything notable about their time in the hospital? Most patients said it was fine. They were happy with their nursing staff and their clinical staff, and there were no issues. Only notable things were patients who had complications, which is understanding. A patient had, for example, a UTI, had a indwelling catheter for a long time, and is now having issues with continence of their neobladder. So they say that they, think about that time a lot. If there's anything that should have been done differently, is that why I'm incontinent? And then, another patient, which was kind of eye-opening to me, we deal with ileuses all the time. If I tell patients, if you're getting a cystectomy, you're probably going to get an ileus. We just plan for it mentally. This patient was funny. He said, all I remember about the hospital was dreaming about Diet Coke and ice while they had their NG tube in. So patients are really residual. I'm sure you are really strong in what they have to go through. I can't imagine having to. Anyways, okay, so learning to care for their urinary diversion. We asked them, what in the hospital made you feel confident in going home and caring for your urinary diversion? And most people said, having practice. They might have changed it once or twice in the hospital with the ostomy team, but they had a family member there who was doing most of the changes. And the thing that really helped them become comfortable with caring for it was when they got home, managing it themselves, whether that was flushing their neobladder on their own or changing their own bag a couple times and making it part of their daily routine. Okay, long term, so what's it like living with a urinary diversion? Most patients said that it's just become a part of their daily life. They've learned to adjust. Some people had to make more adjustments than others, but they feel like they live a normal life. A lot of the patients with neobladders who might have to catheterize mentioned that their day revolves around catheterization, but they've kind of learned to live with it. Activity, so it was really reassuring hearing how active all these patients are, even though some of them have recurrences of their disease and are currently getting salvage therapies. They say they still live an active life. I walk every day, I live a normal life. Most people's limitations in San Diego was just that they don't surf anymore or go swimming, but they've learned to kind of live with that, which was great. This one really surprised me, actually. So when it came to sexual function, most of the men reported, having no erections post-operatively, but they said that was a surprise to them. They did mention that, okay, my surgeon probably brought that up, but I wasn't really thinking about that going into a cancer surgery, but they were just surprised at their lack of, any erection. All of them did mention that it's not a big deal. They found other ways to be intimate, or they're not sexually active. However, they just were really surprised by that. So, just a reminder that there's so much going on before surgery, but these things just have to kind of be reinforced to the patient who has so much going on. And then another interesting thing that came up was a patient who had a total hysterectomy as well as an oophorectomy, and that put them in menopause. Functionally, they didn't have any sexual issues, but the bother came from the fact that they were incontinent and they didn't feel comfortable being with a partner while they had overnight incontinence, which really kind of, there's more to it than just hormonal and like vaginal length and things like that. There's a lot that goes into feeling sexually healthy, which was highlighted. Okay, emotional mental impact of bladder cancer. I stupidly asked patients, so was there an emotional or mental impact of your bladder cancer treatment? Yes, there was. But more interestingly was how they dealt with it. So a couple themes emerged. A lot of patients said they started to feel normal or, like they had less of a mental toll once they started getting back to their normal life, doing their normal things, being kind of taking care of themselves. Other patients said reaching out to mental health counselors was very helpful. Having a supportive family or spouse was also very helpful. And then I thought another really beautiful thing that came up a lot was the importance of a support group. So meeting other patients who have gone through a similar thing was really helpful. Patient mentioned how they had an instant bond with people they just met because they have gone through this really unique experience, and how that really changed how they live with their urinary diversion. Impact on body image, there was a spectrum of responses. Some people said there was no impact at all. Other people said looking in the mirror, they didn't even recognize themselves. They didn't know what the stoma would look like, things like that. And then how they're coping with it also kind of varied as well. Some people just had to make changes to their wardrobe or things like that, and other people are, still having issues coping with the changes that their body goes through after bladder cancer treatment. So then we asked advice that they would give other patients or what they wish they knew from the start. There was also a lot of gems, which we'll hopefully dive into more once we publish a manuscript, but real quickly, some important things as a clinician that stuck out was patients mentioning bringing things up to your medical team if they have any doubts, which really highlighted for me the importance of creating a space where our patients can be vulnerable, because if my patient has a doubt in their treatment or even in the treatment I'm going to provide, we want to create a space where they can bring that up and we can help them work through those doubts or get them the resources they need to feel comfortable with what we're going to do. Other patients, most all patients said, I want patients to know that they're going to be okay after, they're going to live a normal life. It is a big adjustment. Other people stated the importance of communicating with the patient and also finding support. Support groups, support in their family and friends. And then another thing that I think was important to highlight as a clinician is a patient says, you're overloaded with the whole scenario, you have all this information coming at you, and you might be hearing fatigue and incontinence, but you don't know what that looks like. And once you're home and the Foley's removed, it's a surprise. So it just highlights, I have a spiel that I say before surgery, and I know I cover all of these different things, but it's just a reminder that you might be saying it, but patients aren't hearing it because of all the different things going through their mind before bladder cancer treatment. So that at all phases of taking care of your patient, it's important to bring these things up so that they know what's normal and what's not and what to bring to your attention during their recovery. And then real briefly, so the last part of our study was translation of a decision aid. There's currently only one written decision aid that helps patients decide between types of urinary diversion when going into a radical cystectomy. It is only in English and there's no Spanish translation. So what we first did was we modified it so that the reading grade level and the ease of reading, or it had an easier readability score so that it could be more widely applicable to different patients. We then had 2 Spanish-speaking physicians or clinicians translate it into Spanish. We then took those 2 separate translations and reconciled it into one, a third a Spanish-speaking clinician did that. It was back translated into English compared to the initial English translation to make sure there were no mistranslations, and we're currently sending it to different Spanish-speaking urology patients for review to make sure it all makes sense and it's understandable. Once that's done, we'll be able to pilot test it and potentially and hopefully be able to publish it by early 2024 for use in clinical practice. So a lot to summarize, but in short, we won't be able to understand disparities that relate to ethnicity or cultural disparities until we're able to better involve these patients in research. And then also, we learned so much from the patient interviews, but I think the main thing is that supporting your patient pre-op, peri-op is just as important as their long-term support. So getting patients those resources is important. So thank you to all of you for listening to me ramble about this. A special thanks to all the patients that participated. Beacon for helping us get this project off the ground. My mentor, Dr. Salmazi, who we're accidentally matching today. All of the patient advocates and our research assistants that helped. So thank you.
Very nice presentation. I have one question and one comment. Let's start with the comment. Being married to a Spanish lady from Spain, brought up in French, over a couple of decades now of experience, I've seen time and again that people speaking language will speak with an English speaker, and then I'll be told, oh, they don't want this, they didn't understand that, they don't want this and this. Then you come in, speak even with people sometimes who are fluent. People are scared at the time of their diagnosis. They kind of lose their skill set. They're just, and simply having people who speak, and Spanish is such a huge language culturally, is so important. So I think it puts the emphasis of having people really speaking. And French Canadian, for instance, are really fluent in English still. When it boils down to the conduit, to those nitty-gritty of the details, they really prefer to have the conversation in French. That's really. And then the question was whether when you approach those patients, and you said that unfortunately it was a little bit disappointing in terms of response, were these approaching by emails in Spanish? Was this exclusively in Spanish, or was it that you sent an email in English and then sometimes, unfortunately, no one answers?
We initially approached them by calling them. One of our clinical research assistants is a primary Spanish speaker, so she would call them. And then also the emails we sent out were in Spanish as well.
Okay. Nice work, and certainly worth furthering.
Thank you.
Nice work, Ahmed Mansour from UT San Antonio, and such work is very needed in patients in populations where the Latino percentage is high. My question to you is, what are your plans on coding the qualitative data that you're going to get from the interviews, and how are you going to incorporate that in addition to the survey data, which are already validated? There are many ways of coding this free text that you get from interviews, and I would like to hear your thoughts about that.
Yeah, so there's a lot of data on patient interview and focus group analysis. So what we have the free text data, and there will be independent reviewers, 3 of them, who will go through that data and find thematics, and then that will be kind of combined. And so those are just themes that popped out to me and my mentor, but there's a systematic way to review it, which is how we'll do that.
And more recently now, there is an AI-generated system that can help instead of having...
Yeah, exactly. Yeah.
Good work. Nice work.
Oh, thank you so much.
Hi, Armin Smith here. I thank you so much for the very insightful presentation. I have also a question and a comment. So the question is, it seems like there is a little bit of a theme, these patients were not really good responding, they were not interested in one-on-one interviews. So having done this, what do you think is the best way to engage our Latino patients for clinical trials? Is there something you think that is more effective, less effective?
Yeah, I think, the patients that we did involve, it took a lot of effort in the sense of repeat communication with them. So calling them, they say, I'm interested, call me back later. So just kind of a lot of man or woman power in terms of just following up on these patients who live busy lives and aren't going to follow up to an email on their own. And then I think a really big tool will be recruiting these patients in person. A lot of these patients are a few years out from their cystectomy, so their follow-up in a urology clinic was just annually. So I think just kind of coordinating that was hard just with our limited resources. But I think in-person recruitment will be a big deal. And what one of our patients said on the panel, kind of something that we'll probably try to do more, is reach out to the community. In San Diego, there are support groups for, like Spanish-speaking patients with breast cancer and things like that. There wasn't one necessarily for bladder cancer that I found, but I'm hoping in different communities that might exist, or, through churches, things like that. So kind of going into the community more to recruit patients.
Yeah, and the comment I had was, I think it's not just a language barrier, I think it's also, a lot of it's very cultural. I've noticed in patients that I've taken care of that were of Hispanic descent, there's a lot of trust in the doctor, and there's not a lot of dialogue back and forth. They just want to hear what you recommend, and they want to, go with it. So I feel like every time I try to enroll a patient in a trial, I feel like, are they doing it to please their doctor, or are they really interested? So, it's just something that we need to learn more about.
Yeah, absolutely. I've definitely had patients where I give them their options and they ask me what they should do. And I'm like, no, that's why I explained the options so you would decide. I don't want to decide for you. So I definitely sympathize with that sentiment. It's definitely something we need to work on.
Thank you.
Hi, I'm not a physician, and your research is very different than mine, but I really liked your presentation. And this goes, I think you've answered my question maybe already. This goes to what Jim was saying when he was on the patient advocacy panel the other day. I have a question about strategies to identify advocates within the community that can help you increase enrollment. What are the strategies you talked about, maybe church and other community groups, but how do people that do your type of research strategize to achieve that?
So I'm obviously very early in this type of research, so a lot of it has just been people who know other people. So clinicians or practitioners who have been doing it for a long time tend to know people in the community or resources. So for me, it's really been kind of working off of people who just have a lot more experience than me, and those people might be able to answer that question better. But yeah, it's been a big learning curve for me.
But I would think, once you have those people and you have that network, as your research goes further and deeper, that's going to be really valuable for you.
Yeah, hopefully.
Thank you.
Thank you.
Thank you for the wonderful presentation. I think it's a really very, very important study, and I think it's very important that you're kind of this independent third party asking these kind of questions. I've been a Beacon survivor to survivor volunteer for the last couple of years and speak to a lot of patients, and I can tell you that they share their fears, concerns, and questions with us that they would never share with their surgeon. And I just hope that when the study is finished, that maybe we could arrange to have you come and present the results to the group of survivor to survivor volunteers. I think it would give us a really important perspective.
Yeah,
that will help us.
I'd love that. Reach out to me on the app.
Okay.
Yeah, great.
Let us know when it's finished. Thank you.
Thank you.
Hi, Ava. Very nice work.
Thanks.
I'm Karen Sachs. I'm a patient advocate as well as a nurse navigator, and several years ago had a Spanish-speaking patient and had very few resources for them. Thankfully, Beacon has now translated many of their resources into Spanish. I also want to just put a plug in that when your decision aid gets translated, please put that out widely, because all of us really need that.
Yeah, absolutely.
And I also want to say I think you're on the right track in terms of trying to get these patients who are Spanish speaking to them in person, taking advantage of when they are in their clinic visits to, to speak with them. And I find that for this population, needing to have some patient community advocates for them to really kind of walk them through the process and help them understand at a very basic level is something that's needed as well.
Yeah, absolutely. Thank you so much.
Hi, I'm Bob Schreiber, also patient advocate, survivor to survivor participant. I thank Doug for his suggestion that you talk to us as a group. And I have another thing that I'd like to volunteer you for.
Okay, great.
Which is Beacon has a survivorship task force, and everything that you presented matches very well with many of the subjects that we've talked about. So after this session and when there's the get-together session, if you could maybe either see me or, more importantly, talk with Mary Dunn as well as Christina Amodi. They are the co-chairs of the task force. I think it'd be really great to have you as part of that.
Yeah, I would be honored to help in any way I can.
Good,
so
good.
Thank you for bringing
And you're
that
going
up.
to Tennessee, which is the next state over to North Carolina. That's where Mary is, so it can't be too far away.
Okay, perfect. Yeah.
Yeah, we come from Boston. Every state is close.
Yeah.
The other thing I wanted to sort of ask you about is, ask a question is, somebody mentioned AI as helping out, and certainly over the next few years, we're going to get into the realm of having instant communications between people of different languages. How do you think that's going to improve the situation, or are there still, truly significant barriers, like in terms of translation of, written documents, and even with the spoken word, are the cultural and societal barriers still so significant that we'll still need to have somebody who really understands the language?
Yeah, I mean, I think that's a whole talk on its own, but a lot of the issues with AI is that it doesn't understand yet, or I don't know if it will, cultural, societal experiences, things like that. So I did, just for my own curiosity, put decision aids into different AI translators, and then when reviewing it, it was either it just didn't make sense. It either directly translated things, didn't conjugate tenses correctly. I imagine all of that will improve over time, or didn't kind of get the spirit of what was trying to be said in that sentence across. I imagine that'll improve over time, but I like to optimistically think we'll always need the human touch to kind of influence patient aids in terms of making it understandable for that patient, despite their language or cultural background, things like that. So I think it could help, but I think we'll always need patient and clinician input.
Okay, thank you.
Mm-hmm. Thank you.